There are a few stages in DNA replication to ensure that we have an exact copy of the DNA. Okay, the first step is that the DNA is unzipped with an enzyme called helicase. Okay, just remember that the DNA molecule itself has hydrogen bonds running between the bases. Helicase, this enzyme, represented by this arrow, breaks the hydrogen bonds between the A, T, C's, and G's, okay, the bases. The second step is that the two strands separate, okay? They separate because those bases are no longer bound to one another. They're not complementary base paired. The third point here, or the third step, is that the free nucleotides line up alongside the unpaired bases on the template strand. Okay, because those bonds were broken, these DNA bases here are now free. They're not bound to anything. Inside a cell, you have these things called free nucleotides. Okay, these nucleotides are just floating around and represented by these red T's here with the bases. These will complementary base pair to the template strand, the blue strand, and then start to form new DNA molecules. So new nucleotides are joined together with an enzyme called DNA polymerase. Okay, it basically ensures that we have one continuous strand of DNA. The last point here is that we have two exact copies of DNA. One of them, the blue one, is going to be our original template strand, and then we've got our newly produced red piece of DNA, okay, which is going to be from our nucleotides that were base paired to it. If you can see AT, AT, TA, TA. Okay, all these bases are exactly the same in both strands, therefore we have an exact copy of the DNA. This is another way of looking at it. Okay, it's a bit more complex than what we've just been talking about. We have something called a leading strand and a lagging strand. So what happens is that when the DNA replicates, remember we've got an enzyme here called helicase. This is breaking these hydrogen bonds, and the DNA is unzipped in this direction. We have something called a leading strand, okay? The leading strand is going from a 3' to 5' direction, that left-hand side of the DNA molecule. We're going to have continuous replication here, or continuous nucleotides which are being added, and they're added onto the 5' end of the new DNA strand by the polymerase enzyme. On the other hand, on the other side of the DNA molecule, we call this the lagging strand, okay, because we're going from a 3' to 5' direction. We can only add the molecules, or the new nucleotides, from the 5' to the 3'. Therefore, we have to start here and move in this direction. Okay, these are called Okazaki fragments. They're produced in steps and then bound together with something called ligase, which binds the one strand together.